It's an honor to be a part of the fifth Minimal Cell virtual workshop. This is my first um, uh, I could join the minimal workshop, and my talk focuses on solar energy driven photochemical synthesis of proto-cell model Jivanu, which shows ATPase-like activity. And this work intended to explore how the sunlight powered the earliest steps of chemistry to biology. I have been associated with the abiogenesis and was working with the problem of origins of life. And while conducting these experiments, we realized that the photochemical reduction of carbon dioxide into organic carbon and photophosphorylation reactions are the basic mechanism to transduce sunlight into some energy-rich compounds. And my teacher, he started conducting some experiments in prebiotic condition in laboratory simulated atmosphere. We started with some nitrogenous salts like ammonium molybdate, diammonium hydrogen phosphate, some inorganic minerals, and formaldehyde was the source of organic carbon. We prepared sterilized aqueous mixture and exposed this mixture to sunlight. So these efforts are entirely different from the I have seen. I must congratulate John for the excellent work which has been initiated for the minimal cell efforts. And it was a great enlightening experience for me to attend this minimal cell activity. I have got a lot of insights. So these are the mixtures which we have prepared and started. This is an unexposed mixture. And when we expose the mixture to sunlight, immediately it is a ultra fast reaction. I have also worked with ultra fast laser flash photolysis experiments. I'll let you know later. And when we continue our exposure to light, it becomes dark blue. It shows the precipitation of certain organomolybdenum microstructures. And these are the systems which are getting synthesized in the solution. They are spherical in shape, bluish in color, and have a definite boundary wall. And you can see that these microstructures, this is an optical microscopic image showing the multiplication by budding. They grow by the photochemical synthesis of material. And we can see the two regions. Outer is the limiting boundary. Another image clearly there is an intricate internal structure is there. And the smaller unit comes from the larger one. When the exposure is increased, and we have also taken the scanning electron micrographs of these organomolybdenum microstructures. We can clearly see that these are the budding vesicles. Another image. And it was interesting to observe various images are there. And this is the electron transmission electron micrograph. And when we have taken the cross section of the structure, clearly shows the definite boundary wall and some central structure. This is the elemental composition of these organomolybdenum structures, which were photochemically precipitated in the picture. FTIR. The purpose is just I want to show that we expose this sterilized aqueous mixture to sunlight, and this mixture contains some inorganic organic substances. It has shown the abigenesis of various materials. More than 12 amino acids have been reported and some small peptides. And this work is in progress since last more than 4 decades in India and in some places abroad. These are some publications related to on photochemical synthesis of amino acids and some small peptides. And around more than 50 students have earned their PhD degrees and papers. These are some papers related to the abigenesis of nucleic acid-like bases in this mixture. They have purines and pyrimidines as well. And we have what's interesting to observe that some that this picture shows some enzyme-like activity in this picture. There is some publication. We were having some collaboration with Ames, NASA, and Laird Folsom and Adolph Smith. They were working with these systems, and they could observe the past acetylene overhead this mixture. And this acetylene was reduced to ethylene and finally to ethane, indicating the presence of photochemical fixation of nitrogen and the presence of nitrogenase enzyme in the mixture. And so after in the subsequent experiments, the nitrogen was labeled with N15 and the carbon was labeled with C14. And it was found that this hydrogen evolved in this mixture by photolytic decomposition of water, and it is utilized in photochemical fixation of nitrogen and oxygen, because the acetylene became CHD. I was working with the ATPase enzyme, and I was some lab in France. We are working with Dr. D.A. Lerner, and we worked with labeled ATP and labeled AMP, and we could discover that negative mixtures did not show any enzymatic activity, but other mixtures which were incubated for different incubation period, they showed the presence of ATPase activity in the mixture, and they were counted by Beckman counter, radioactive counter, and some more experiments were performed. And the UV visible spectra of Jivano mixture. This is the control mixture, and this is the absorption spectrum. Control line. Sorry. Some proton pump experiments were run just to see the absorption in the mixture. This was done at the Waterloo University. Professor German Ciani was working with us. And this is the plot showing that how the budding vesicles, they increase in size. When the exposure is increased in time, the size of particle also increases. So ATP and ATPases, they are energy transducing, energy producing molecules in the system. And the interesting thing is that ATP is universal in all living systems. ATPases, including all ATPase synthetases, they catalyze ATP formation and hydrolysis, and they play central role in energy conversion, metabolism, and origin of life. There's some references showing the enzyme-like activities, and this was the detail of the experiment when we were conducting some experiments for the ATPase. So seems that ATPase synthetase, it is universally conserved in nature in various domains of life. It may be bacteria, archaea, and eukaryotes, and there is a, and these molecules, ATPase, they are the ancient molecular machines. They transduced energy. All energies can be traced to sun, and these ATPases, they help in the proton pumping and the sodium gradients, all mediated by ATP system. So these ATP studies, they have enabled us that this biogenic system, metabolic networks, and cellular complexity, how we can study and pursue. So in prebiotic system, the most interesting molecule is the acetyl phosphatase. It leads to the production of ATP finally. And one thing I forgot to tell, that I have introduced different level of oxidized sulfur, sulfate, sulfite, thiosulfate, and sulfite. And I was working in King's College with Professor T. Hall. It was long, it was around 84 at King's College, and he could detect pyridoxine-like material in all the 7 samples I prepared. Pyridoxines are the iron sulfur protein where ferric ions are ligated to cysteine of sulfur, and these ferric ions, they also help in the catalysis from ATP to ATP conversion in water. So ATP favored the formation of triphosphates in the mixture, and ATP became universal. It also indicates that the similar systems like Jivan existed in prebiotic era, which converted sunlight into energy-rich compounds. Professor Clarence Smith, he has mentioned in his book Genetic Takeover that Bahadur and his group, they have been studying the microstructures, complex budding vesicles that Bahadur calls Jivan, which were precipitated in sunlight with ammonium molybdate, diammonium hydrogen phosphate, some minerals, and formaldehyde. And the synthesis of number of biochemicals have been reported. And he concluded that in some earliest energy transducing system was possibly a similar grade of organization to Jivanu, which had the capability to convert sunlight into energy-rich compounds. So this is what I've been intending to submit that we are having a system in definite limiting membrane for charge separation. We have a system of iron sulfur protein, that is pyridoxine, which can mediate electron transfer. And we are getting the evolution of hydrogen in the mixture. Of course, it is in a little amount, but it is there. And it has been confirmed by the photochemical fixation of nitrogen and carbon dioxide. So it is a catalytic system. And the photochemically synthesized proto-cell like Jivanu. A system like this can mimic ATPs like activity. So what is the minimal? My point is, what is the minimal threshold of complexity required for a chemical system to be recognized as alive? Is it replication, metabolism, or the ability to harness energy? So it's a different kind of work which have been presented in this meeting, where it is just a summation that we have basic components and some more effort, more experiments are to be conducted. And it will be very interesting to seek your valuable suggestions and guidance so that we can plan some experiments in collaboration if possible. So thank you very much, sir.
Thank you. Thank you, Vinod. Thank you for introducing me to, and your colleagues here, to a system that I knew nothing about. I know I try to do reading on non-living synthetic cell systems, but this is one that I was unaware of.